What we're doing is trying to take mutations in different genes and combine them together to understand how genes work together to coordinate cell life. And we do this in a simple organism like yeast that only has 6,000 genes. But remarkably, yeast is very similar to humans, so it provides a very good model to understand. So what we learn in yeast can provide us with some direction or a reference to understand, to help us understand how genes work together to modulate essential functions and life in human cells. So what we do is take a collection of yeast strains, each of which is deleted for a different gene or mutated for a different gene. And we use a technology we developed in our lab that allows us to combine pairs of mutations in the same cell. And then we can simply ask, do mutations in these pairs of genes allow the cell to live or not? And we use robotic technologies to do this, and we were able to test all 18 million possible mutant combinations. And we don't think that genes work in isolation. So there are obviously many examples of Mendelian diseases where a single mutation in a single gene results in a disease. So those cases would be highly predictive. We think that many common diseases are affected by combinations of mutations, rare mutations in different genes. What we've done is tested all possible double mutant combinations to build this large network, and we see many structures in these networks that we think could provide a reference from which we can start to learn rules and principles that might help us to understand or simplify the problem in humans and help us understand develop rules of how different genes interact. We believe that this will help us simplify the problem, or at least provide a guide to start to understand how genes work together to modify complex disease in humans.